Hello everyone, I am Sutapa Datta. Welcome to my channel Sutapa's Biology. My today's topic is structure of the gametes. Structure of the gametes means the structure of sperm and structure of ovum. Before that, I am telling you that you will get all the diagrams of my previous videos in my description box. So follow the description box regularly, and another thing is that just watch the complete video, and up to the last of the video if you watch, then you will get many important informations for your board exam as well as your competitive exam. So let's start today. That's the structure of the gametes. Structure of the gametes, and that is the firstly this has two division, the structure of ovum and structure of sperm. Number one, structure of sperm. And number two, structure of ovum. So firstly I am starting the structure of sperm. The sperm has four, the complete form is looking like the tadpole. It's tadpole shaped body with a tail or flagella, and it has four parts. In the complete structure, you can get the four parts of the sperm, that is the head, neck, middle piece and tail. So let's draw the structure. For the diagram, I am just omitting this one. You know that this is structure of sperm, obviously under microscope. This is the cap of the sperm. It's very smart. It has a cap also, and this cap is known as acrosomal cap on the anterior part of the head. And within the head, main thing is the nucleic acid or the nucleus. The main thing in head, it has dense nucleic acid. That is the DNA or nucleus. It's known as nucleus also. This is the head. Next one is neck, and neck is consists of two centrioles. One is proximal centriole and another is distal centriole. On the basis of the positions, they are divided into proximal and distal centriole. After that, a very important thing in the middle piece, that is the ribbon-like mitochondria here, spirally coiled. Very thick association of the mitochondria. So many mitochondria are present here that they coalesce or fuse to form the ribbon-like structure. many mitochondria are required in this structure, as sperm is a mobile structure. It can move well and it can swim quite fast. So that's why they need the maximum amount of energy, and that's why mitochondria are many in number. As many in number in a small place, that they coalesce with each other but functions well. Now, there is a thin layer of the cytoplasm, which we can see in the middle piece. That is, this is the layer of the cytoplasm, and after that the tail is starting. So we can divide the total structure in 4 successive phases. That is head, 4 successive structures. There is head, next part is neck, next one is up to flagella, this is tail. Sorry, middle up to this is middle piece. And then this is the tail. So 4 parts are there. And what's about the other thing? That this is the acrosomal cap. Acrosomal cap. I am coming in details later about it. Then it's the nucleus. Then this is the proximal centriole. This is the distal centriole. This is the ribbon of the mitochondria, is known as Nebenkorn sheath. Mitochondrial ribbon. Ribbon. It's in other words is known as Nebenkorn sheath. Nebenkorn sheath. And this thin layer of the cytoplasm just surrounding the Nebenkorn sheath is known as manchette. That is the red lining of the middle piece. Manchette. So after that, the tail, in the tail part, this is the flagella. So this is about the total structure, and about the different functions of the different parts. That is, head is the main part, which is provided with the acrosomal cap, and this acrosomal cap, the anterior part of the acrosomal cap has the main function, and that's known as gallia capitis. The anterior part of the acrosomal cap is known as gallia capitis. This is very important, as in this part the main lysing enzymes are present, like the main lysing enzyme in hyaluronidase and lysine, etc. Hyaluronidase is the main important. Hyaluronidase is the main sperm lysine. Acrosin is also there. These are required for the penetrating the ovum wall. Now after that, this one hyaluronidase and acrosin, that is after, during fertilization, the acrosomal cap get dissolved to release these enzymes, and this will help to dissolve the ovum wall that is made up of hyaluronic acid. Hyaluronic acid will be dissolved by the enzyme hyaluronidase. So acrosomal cap is very important for the sperm for its function. And next is that it has the nucleus, and this nucleus is the main thing that contains the 23 chromosomes which takes place in the fertilization. And during the fertilization, this head of the nucleic acid or nucleus of the sperm, as well as this one, that is the proximal centriole, goes inside the ooplasm within the ovum. Why? Because nucleic acid will do the division to form the zygote, and this centriole will take place in the division. In the division of the zygote, the proximal centriole take part. And what's the role of the distal centriole? It forms the axoneme. The flagella has the axoneme, that is the central axis, which is known as axoneme, and that's formed by the distal centriole. So this is the function of the neck. The sperm neck has two centrioles. One is for division phase of zygote, and another for formation of the axoneme or flagella. Now next is that the middle piece, it has only the very thick association of the mitochondria to provide maximum energy to the for the fertilization, and another is the manchet, which is the cytoplasmic thin layer. And after that, the tail has the first part is covered by some cytoplasm, and last one is just naked axoneme, and that's the flagella, and it helps itself to move the sperm. So this is about the structure of the sperm, and later in this video I am coming to the structure of ovum. Now coming to the structure of ovum. Already I have completed structure of sperm. Now structure of ovum. It's a very simple structure. Round structure. It's non-motile structure. Structure of ovum. It's a non-motile round structure. No complexity is there. Just it has this type of round center part, which has dense ooplasm, as it will provide all the nourishment, initial nourishment to the zygote. So it has very dense ooplasm. At the side of the ovum, there are three degenerating polar bodies. In my last video, I have told you about the rules of the polar bodies and formation of them. So these are the degenerating polar bodies, and this is the nucleus of the ovum, which is known as pronucleus before fertilization. Up to fertilization is known as pronucleus. So, and the layers of the ovum are like this. This is known as vitelline membrane, the innermost one. Next is zona pellucida. I am going to discuss about the role of these layers also in this structure. This is zona pellucida, and just outside of this, there are many radiating cells which are not so compact. Those are almost fragmented cells radiating in nature, radiating from the outside, not giving all the cells. It's completely surrounded by this, but I am giving only some cells here, and these are known as cells of corona radiata. So this layer is known as corona radiata. So outermost one is corona radiata. It has the cementing material, that is the hyaluronic acid. So fertilizing sperm has to first cross this wall. First hurdle is crossing the corona radiata. Actually, during the fertilization, the sperm has many challenges, as it has to compete with millions of other sperms. So for a single ovum, millions of sperms will compete with each other. So it's a very tough competition. You just think, more than the NEET also sometimes, isn't it? So this is the corona radiata, and after that the next layer is zona pellucida, this layer has the very important role in implantation. So when I shall give the video of the implantation, then I shall tell you about the role of zona pellucida. And after that, this is the vitelline membrane. So firstly, the sperm has to meet this coronal radiata. Then if it many sperms, 5-6 sperms can cross this wall, and after that they have to cross the zona pellucida, and lastly vitelline membrane. After crossing the vitelline membrane, there will be the formation of the fertilization membrane, and that will prevent the polyspermy of the gamete, or polyspermy of the zygote. Now this is the vitelline membrane and this is the nucleus. So this is about the structure of the sperm, and these are the degenerating polar bodies. They have no function. So these are the degenerating polar bodies, and this is the ooplasm, dense ooplasm. So this is about the structure of ovum, and it's very simple structure in comparison to the sperm, and it is non-motile. So in fertilization it has no such role, but after fertilization, from the formation of the zygote, there are lot of functions. And in my last video I have told you that during oogenesis, up to the secondary oocyte formation takes place before fertilization, and after that, after completion of the fertilization, if it's get fertilized with the sperm, then it will form the zygote by completing it meiosis II. This is about the structure of the sperm today. So this is about the structure of the sperm and ovum today, about the structure of gametes.